Hello there. I'm going to go through the dichotomous key slideshow. I don't usually have a whole lot of time to run through it during lab, and it's something that we use quite often in the lab part of the course. And so I'll have it here for your reference. On this slide, I have a sample Burgess chart. It's a lot simpler and shorter than the one you will see for most of our assignments, but this is good for a practice. So instead of 24 some starting organisms, we have just 10. So there are 5 gram-positive organisms here and 5 gram-negative organisms below. The sample we're going to use today, we're going to pretend that we have been assigned Shigella dysenteriae. All right, so in most of our assignments, you don't know what you've been assigned. You've been handed a broth and there's something cloudy inside and you're told to start to figure out what it is. In our first assignment, called Burgess assignment, you're given an organism. You're assigned an organism that you know, and the exercise is to go through and construct a dichotomous key for it. So this is going to follow that type of procedure. This is a really busy slide, and we're going to spend quite a while on it. So this is how the dichotomous key is set up. Here at the top of the key, you see all the starting organisms. Right? So if I go back to the previous slide, you'll see that you were given this chart, and all the possible starting organisms are listed here for you. Now, granted, that's not a terribly realistic case, but hey, this is microbiology, first time through. All right, so all our starting organisms are here. Now, what I've probably said before is that we do pay attention to the notation or the format. So notice this first time that they're listed, all the organisms are listed together. It is not necessary to put anything in bold like I have here, but for this Burgess assignment, I know I have this, so I can put it in bold. Normally, you wouldn't know that until you get to the end of your assignment. All right, so they're fully listed, genus and species name. This is a computer-generated printout, so I have used italics. Okay, so genus is capitalized, species is not capitalized, and it's all in italics. You have the choice to do it in computer, or you can write it out by hand. If you write it out by hand, you cannot fake italics. You need to underline, even though if you have a slant to your writing. Yes, you need to write it just like this with the same type of capitals and small letters, and then you need to underline every single thing that is italicized. All right, so all possible organisms listed at the top. The next step is that we usually separate things by morphology. Now, morphology is shape or appearance. And there are some do's and don'ts. In general, we do always separate by gram type and shape in our projects. There's exceptions to this. There may be a project where you've been told all of our starting organisms are gram negatives. In that case, you wouldn't need to do a gram separation step. Okay, but I'll take it from the top. We often do separate by gram type and shape, and we do it at the very beginning if we're going to do it. There are other morphological tests: arrangement, color, capsule. Let me see if I've listed it. I sort of abbreviated this. If you look at the full Burgess charts, there's shape, arrangement, gram stain. There's capsule, there's motility, there's arrangement, there's pigment, at least. So those are morphological characteristics, and we do not use them on a dichotomous key. That is, we don't use them for true identification. They are not reliable. They're subject to handling, to culture conditions, so we do not use them. All right, so gram first, usually. I make a separation bar like this, and my test is gram type. There are 2 separations. It can be gram positive or it can be gram negative. Now, I know what they are because my chart gives that to me and I can easily look that up. So I separate them by gram type. Notice that the first time the organisms are listed, the full genus is spelled out, but after that, you can leave it out. This would be helpful if you're writing it out by hand. If you're doing it by computer and you just copy paste, it's okay to leave the whole unabbreviated form there. All right, so I've got them listed like this. Now, I know I have Shigella dysenteriae assigned to me. In another type of project, you would actually carry out a gram stain and you would see, let's say, pink shapes. You got pink shapes, you're going to find out that's gram negative, so you know you have a gram negative. What you do is you eliminate what you don't have. Right? So I don't have a gram positive, I eliminate them from the key. I like to use the elimination, I like to show the elimination, and I'm going to ask kindly that you do so also. It just makes the key a lot clearer to read through. I eliminate them, but I don't scratch them out so that I can't see anything that's under there. It's helpful to see what's under there just in case you want to go back and check out some things. All right, so eliminated like this. The next test is going to be shape. So same thing, a bracket in two parts with the two different shapes that I could have. I've written rod and coccus. You can say rod and circle. You can say bacillus and coccus, bacillus, circle, etc. Okay, I don't mind. Shape. Again, this is indicated to you on the Berge's chart, or you would have done a gram stain or even a simple stain and seen the shape that your organism is. Right. So Shigella dysentery is a rod. I don't have a circle-shaped bacterium, and so I eliminate that. Now at this point, we have used up all the morphological tests that we can, and so now I have to go to biochemical tests. For biochemical tests, I go back to the Berge's chart, and I look here. Everything after gram stain, here on in, these are biochemical or metabolic tests. They are usually very reliable. So I look at these, and I have 3 remaining organisms. I have these 3 gram-negative rods, and I'm going to look through here for a biochemical test in which those 3 have some differences. Okay, so let's say this first one, ferments glucose. This is not a good test because they all are positive. A for acid is positive for carbohydrate fermentation. And they all produce gas. This one produces gas sometimes, but sometimes it doesn't. So that's not a good test. Some of these other ones are not good either because we have D's. D is differential, or it means it could be positive or negative. It's not consistent. So that's the same as the blanks here. We would avoid that. Whoops. We would avoid that. Here's all positive. Not a good test. All negative. All right. So you get the idea. Here are some good tests. All right. Methyl red is a good test. Positive, positive, negative. Vogts-Proskauer next door is also good. Negative, negative, positive. All right. So arbitrarily, I pick Vogts-Proskauer. Actually, methyl red is probably the better test. Anyway, here on the chart, whoops, go back to where I was. And I'm going to separate these 3 rod-shaped gram-negatives by a Vogts-Proskauer test. Positive and negative results. And looking at my chart, I put them in their corresponding sides. And I know I have Shigella dysenteriae, or I would have carried out the test. And if I had Shigella dysenteriae in my test tube and carried out the Vogts-Proskauer test, I should get a negative result. And I cross out the side that I do not have. All right, so I'm left with those 2 organisms, Shigella and E. coli. And same thing, I go back to the Berge's chart, Shigella and E. coli, and I look for a test that they differ in. Let's see, ferments mannitol, ferments lactose. Okay, those are both tests that I could use. Oops, there's my mistake. I changed things. This really should be moved over. Okay, so I'm going to scoot it over. Can't do that right now. All right, this really should be scooted over to the Shigella and E. coli. And a negative and a positive side. E. coli is positive. Shigella is negative. And I cross out the side I don't have. And there I have it. I usually put some indication next to the one that I have, so it's nice and clear that's my final identification. Well, maybe not so final. What we often request is a confirmation test. The confirmation test is an additional test that you carry out and you put on your chart. It's a test that should give different results between your last remaining organisms. I mean, you're only at the last step when you have your assigned organism all by itself, ideally against one other organism. Having your organism against 2 others is not as ideal. Against 3 others is even less, because it's difficult to find a test, an additional test where your organism has one result and 3 others here on the other side have a different result. So it's best to end up with this type of situation. All right, so I'm going to carry out my confirmation test. And remember, there was a mannitol test that we could have used. So I have that. I have to write it out, confirmation test. I really like it when it's this way and not just another part of the dichotomous key. I want it really clearly shown that I have proposed and I'm running a confirmation test. This is what it is going to be. It's going to be carrying out a mannitol fermentation and looking for the presence of gas. And here I'm going to clearly list showing that my last 2 organisms have different results. And if I were to carry it out, I certainly should hope I should find no gas in my mannitol fermentation, right? And so I indicate, let's say I actually did carry it out and I got the result. And this shows me and everybody else that with pretty good confidence, I have Shigella dysenteriae. All right, my time is running out, so I'm actually going to do the last 2 slides on a separate little video recording.